1. Please give a warm welcome to our partners and sponsors. Standortagentur Tirol. As part of the Lebensraum Tirol Group, the Standortagentur Tirol works on the innovative future of Tirol. Together with companies, scientific partners and public institutions, we enable innovation, create a stakeholder network and promote sustainable development. We accompany and support with our expertise in the areas of innovation, digitalization, growth, finance and European network. Our mission? We make Tirol the most sustainable, innovative region with a high quality of life. Startup Tirol. Startup.Tirol is the first point of contact for startups in Tirol. Startups receive support from idea to growth stage and benefit from the compression of information, national and international network access and specific expert know-how. If you are a startup in Tirol, feel free to get informed via www.startup.Tirol and contact us for further support. Alpine Tech Innovation Hub. The Alpine Tech Innovation Hub is an international innovation and entrepreneurship centre for alpine technologies. The Hub is a platform for regional and international exchange and innovation promotion by leveraging the existing strengths of alpine technologies. International startups in the market-ready phase can now apply for the Alpine Tech Accelerator 2021, the 3-month support and network programme in Tirol. Tyrolean Economic Chamber and Junior Chamber. At the Tyrolean Economic Chamber, everything revolves around companies and we do our very best for the Tyrolean enterpriser. The Tyrolean Economic Chamber represents the interests of more than 50,000 entrepreneurs. As a strong voice of this audience, we advocate a future-oriented and business-friendly policy. We promote the economy through a variety of services, support Austrian companies with our know-how, and stand for a modern social partnership. As part of the Tyrolean Economic Chamber, Junior Chamber and the Industry Division support the event Idea Meets Business because our members benefit from new technologies developed by startups and startups learn from established companies. Register now. Werkstätte Wattens. Werkstätte Wattens is an international business and creative centre for founders, startups and service providers at various stages of development. Werkstätte Wattens is a partner of the IECT Summer School since the beginning, with the aim to support high-potential technology startups and connect them with the local startup and entrepreneurship community. To find out more, follow their social media channels or visit the website. Infineon. Infineon is a leading semiconductor manufacturer active in hardware security, sensors, connectivity, and power electronics for a broad range of industrial, automotive, and consumer applications. We're a proud partner of the IECT Summer School to support entrepreneurs who develop and commercialize innovative semiconductor topics and related applications. Talk to us if you think that Infineon could be a valuable business partner on your side, a customer, supplier, research and development, or go-to-market partner. Tiroler Sparkasse. Founded in 1822 on charitable principles, Tiroler Sparkasse is the oldest financial institution in Tyrol. From its formation until today, it has always been operating under the premise of creating and ensuring prosperity for the entire region. Throughout the years, Tiroler Sparkasse established itself as a modern and innovative full-service bank. With its state-of-the-art online banking system, George, Tiroler Sparkasse is adapting to rapid digitization without losing its traditional values. We believe in future technologies, entrepreneurship, and we believe in you. Tiroler Adlerrunde. The Tiroler Adlerrunde is a politically independent platform of renowned entrepreneurial personalities from various industries. It combines entrepreneurial spirit, vision, and a deep connection to its Tyrolean homeland. The Round sees itself as an impulse-giving think tank with the clear goal of creating the best possible framework conditions for a strong Tyrolean economy in harmony with sustainable values. The members maintain a constant and open exchange regarding current and future developments and challenges with the involvement of socially relevant areas and institutions. Ludwig Boltzmann Gesellschaft. The Ludwig Boltzmann Gesellschaft is a research organisation with a thematic focus in medicine, life science, the humanities and social and cultural sciences. It specifically initiates new research topics in Austria. Together with its partners, the LBG currently operates 20 Ludwig Boltzmann institutes and research groups. It also develops and tests new forms of cooperation between science and non-scientific actors, such as companies, the public sector, and civil society. Since its foundation in 2016, the Career Centre of the Ludwig Boltzmann Gesellschaft has been pursuing the goal of career prospects of the approximately 250 young scientists of the Ludwig Boltzmann Institutes in and outside academia. Since 2019, selected individual and institutional offers are also open to pre- and postdocs from other research organisations in Austria.
Hello, everybody, and a warm welcome to the 7th ICT Summer School on Entrepreneurship and to our second event, which is for our network. It's called State of AI. My name is Clara Brandstetter. I'm the Managing Director of the ICT Hermann Hauser, and in the name of the overall team, or on behalf of the overall team, I'd like to welcome you to State of AI. Before we dive into today's session, I shortly like to introduce you a little bit to the ICT Summer School, as this event is part of it. This year, it's already for the 7th time that we have the ICT Summer School, and for the 2nd time, fully online. But every time, it's a bootcamp for entrepreneurs, and it's a think tank and inspiration for entrepreneurs, for venture capitalists, for experts, as well as corporates. So it's kind of a deep dive into a deep tech world. And at this year's summer school, we have 35 participants, which is actually quite a lot. And it's again really international. We have more than 17 nationalities. So we have different cultures as well as different backgrounds. So it's a really interdisciplinary program this year. And all these participants are coming from more than 27 universities and research centers. So there's a lot of collaboration and exchange. More than 40% are in the life science area, 60% are in deep tech, means quantum computing, material science, so all really interesting topics. And if you want to get to know our participants and you are like watching State of AI from the ICT network, you can check them out on the dashboard. If you're watching State of AI on YouTube and you want to get to know the participants, then just log into network.ict.at and you will find the participants video on the dashboard. And I can highly recommend to watch the videos and the pitch intros because you will enter totally new worlds and get to know a lot of interesting talents, people, and ideas. So back to tonight. What's going to expect you? It's already the 5th time we're having State of AI, so we are celebrating our 5th birthday, and we do State of AI always in cooperation with AI Austria. And it's actually quite a nice story because AI Austria has been founded whilst the ICT Summer School 5 years ago at the Werkstätte Watten live on stage. We are currently as well streaming from the Werkstätte Watten, so we are kind of the birthplace of AI Austria. And may I welcome one of the co-founders of AI Austria. This is Clemens Wasner. And Clemens Wasner will host this evening. And Clemens, it's a pleasure to run this event every year with you and to dig into the latest topic of AI. And this year, it's all about AI and biotech, as we realize AI is really the new normal in science. And this has been developing over the last years. And as it's our 5th birthday, could you take us a little bit through the history of the State of AI formats we had in the previous years?
Yeah, definitely. Thanks a lot for the nice intro words, Clara. I also hope that you still think positively about our cooperation in 1 hour, but I will do my best. Fingers crossed. Yeah, so AI Austria, or let's say State of AI, started in 2017 in the summer school. I think it was the 3rd summer school back then. And back then, it was more or less the beginning of the AI hype, I would say, in the mainstream media. This is like always like 1 or 2 years, like compared to the tech scene, or 5 years to the ICT. And so initially, it was more like what to expect, in which fields will we see the first applications, where do you see natural limits? As such, we had the various combination back then on a panel. On the one hand side, classical, I would say, IT and hardware people like Hermann Hauser himself or the dean of the ICT department in Cambridge. But we also had a representative from the Human Brain Project, which has a very different approach to it. Yeah, and then over the years, as the topic developed, we also changed, or let's say tried to take this into consideration for the panel planning. So in our second iteration, it was about regulation and trustworthy AI. This was especially interesting because back then the EU asked its member states in 2018 to come up with each member state with the AI strategy. which should serve as the basis for the AI Act. So yeah, lots of discussion back then on trustworthiness. Then in the third year, it was about, let me think about the sustainable, last year, it was about the sustainable development goals of the United Nations. So we also had impact investors here. And to provide us an overview what to expect, let's say, outside of the classical use cases like autonomous driving, or let's say like predictive maintenance or fraud detection, what can you expect in the big wide open world? Yeah, the one I forgot was in 2019. For this, we had the angle on where does Europe stay in this big game being played?
And as well, use cases. I think this one was really about use cases. We had Shiraj Khalik from Atomico and as well from Axel Springer, the CTO, which I think was really interesting to see how it's actually implemented in big corporates and in the media world.
Mm-hmm. And it was also interesting, especially from Atomico. I remember vividly in the panel discussion, the journalist was basically asking a very self-defeating question. It's like, has the train already left the station? Are we only left with an empty bottle? And Shiraj was very quick to point out that for certain fields, like for example, medicine or biotech, Europe is still a very nice place. So it's sort of logical that for the fifth iteration, we do as Shiraj said and make a special panel on this topic.
Mm-hmm. Yeah, but it has been quite a nice development, like with the ethics, regulatory, then coming into use cases, and this year really focusing on the industry. And you already dropped some names. I think we can be quite proud that State of AI always had like really interesting people joining for the panels from IBM, Microsoft, Herman himself, Atomico. So there had always been really good discussion, like controversy and like inspiring as well. That's what this overall format is on. And I'm really looking forward to today's discussion as well, because Clemens, you're hosting it and you're moderating and leading us through it. And we have two keynote speakers, like one is Noor Shaker. She's the founder of Glamorous AI. She will tune in from the UK. And we have Noah Weber, who is the CTO of Solaris Therapeutics. So both of them are entrepreneurs. They will guide us or lead us into the entrepreneurial world of this topic. And then, which I'm really looking forward, is as well the panel, because it's as well international. We have John Cumbers joining us from the US. He is the founder of SynBioBeater. He is an investor himself, entrepreneur, and built up the overall ecosystem. And I think it's going to be interesting to have this US perspective on what we are discussing right now. And then we have Stefan Sauermann joining. He is a regulatory expert and program director at the Technikum in Vienna. So as well, seeing how the regulation is framing the market and allowing us to deep dive into these new technologies. So Clemens, I'm sure our cooperation will stay after this 1 hour, and I'm really looking forward into the discussion. And that's it already from my side. I'd like to hand over to you, and looking forward to the insights we are gaining today.
Great. Thanks a lot, Clara. And yeah, too bad I can't join you later at the buffet. But yeah, the fate of remote events. Yeah, maybe one or two small items for housekeeping. Since this is an online event, we do not take questions directly after the keynotes, but we will answer them over the course of the panel. If you have questions, there are several ways how you can do them. If you're joining via the ISCT tool, it can be done directly within the tool. For those of you that tune in via, let's say, YouTube or on the LinkedIn live stream, you can just write a comment. I think on YouTube, it's on the right-hand side of the stream, and LinkedIn, it's directly to the post. And we can show these posts then directly as questions. This is an example how this will look like. So you would see the picture of the person and the question it's asking. Great. Yeah. So without much further ado, I would ask Noor Shaker to the stage with me. So yeah, hello Noor. Can you hear me?
Yes, I can hear you well. Thank you very much. Hi. How are you?
Perfect. This is the most important part already finished. Yeah. No. Thanks a lot for joining today, and yeah, also to make this happen on such a short notice. Yeah, as I said, we already have your presentation here. I will also put this now here, so you could already move it to full screen.
Cool. So can you see it in full screen now?
Yes.
Cool. Awesome.
So during your talk, I will leave the stage, so it's clear who is talking. And yeah, when it comes to the end, I will join again. So
Awesome.
don't get confused when you suddenly see a second face. Yeah.
No worries.
Thanks a lot, and the stage is yours.
Thank you very much, and thanks again for the invite. So I'm going to be talking today about a topic that is, I think, quite relevant for this event and also very close to my heart, which is basically discovering novel chemical entities and novel drug candidates using machine learning, and specifically within the context of what we do at Glamorous AI, the company I started about a year and a half ago now, focusing on applications of AI for small, sparse, and noisy data sets. And I'm going to talk a bit about why this is important and why we believe this is the next kind of milestone for AI to really kind of be a mainstream in AI for drug discovery. So just a bit of background about who we are and what we do. So as mentioned, Glamorous is about a year and a half old. We are a company here in the UK with a headquarters in London. We have a unique approach to drug discovery with a focus on solving data challenges in AI for drug discovery. And we do that through basically working internally on drug discovery programs, but also we believe that a big part of really bringing AI to drug discovery is around democratizing access to this technology. And we do that through our platform that we call Rosalind AI, which is basically an AI drug discovery platform for the enterprise. It's a first of its kind because it contains the state of the art, but also our own proprietary technology to solve data challenges. And it covers the preclinical stage of drug discovery, so all the way from hit identification to a preclinical candidate. We've run it now on a number of drug discovery programs, and we have wet lab validation on a number of programs that we run either internally or with our partners. A big part of our mission and what we believe is crucial to really solve drug discovery and bring cures to patients is being able to collaborate and being able to put this amazing technology in the hand of drug hunters and experts. So we have a very collaborative approach to how we work with our clients and our partners and how we build our business model. We work with at the moment with a number of leading research organizations, academic institutes, and also leading contract research organizations and small and mid-sized pharmaceutical companies looking to integrate this technology with their own drug discovery programs. Again, our ultimate goal is to solve data challenges, and by solving that kind of big piece, we believe that AI can really be more fruitful and can be really integrated within the mainstream in drug discovery. So for those of you who don't really know a lot about drug discovery, like small molecule drug discovery specifically, it is just kind of one slide that illustrates exactly what we do. So on the right hand, you see a protein structure or like a 3D model of the protein. And in our body, there are an estimate of about 180 different protein structures, and they interact obviously with each other, which triggers a lot of different functions, which keep us alive really. But they also integrate with small molecules, and those are basically those chemical structures that you see on the left hand side of the screen. So these are small molecule inhibitors or activators. They interact with different proteins, and by doing that, they change the function of that protein, and then they change different pathways, and they either cure diseases or they cause diseases. And the main core in small molecule drug discovery is really to find a way to design those novel chemical entities, those novel drugs, novel potential chemical candidates, in a way that makes them unique. That's kind of from a commercial point of view, because as a pharmaceutical company, you would like to patent that drug and you want to take it to market with exclusivity. But also in a sense, from a biological and chemical perspective, you want that drugs to be effective, to be non-toxic, for instance, doesn't have any side effects, and interact only with that protein in a meaningful way, in a way that you are expecting. So it's really kind of a combination of a search problem, so searching for that or designing that small molecules, and it's also an optimization problem from a sense that you want that small molecule that you designed to hit usually hundreds of different properties to actually go all the way from the point of discovery to actually being put in patients. So you need to kind of tick all those boxes before you actually be able to test it in patients. And this is a very challenging and exciting search problem because the potential drug candidate space or the potential space of chemical that could potentially become drug is really huge. It's estimated to be as big as the number of atoms in the entire universe. So it's really a very, very big search space to just maneuver in and be able to kind of, you don't really expect to randomly search that space and tap into interesting ideas for chemistry. On the contrast of that, the chemical space that we humans have designed and have screened against targets is really just a very, very tiny fraction of that chemical space. So we need an effective way to go beyond existing screening libraries into the whole drug-like space. But it's not just about having access to that drug-like space. It's also about being able to identify the subspaces in that huge space that are relevant for specific disease and for specific disease areas, and that are also relevant for us humans. So ideas for kind of those kind of methods that can come up with those novel chemical ideas and novel drug design came initially from basically other domains, specifically from image and text processing. And these are basically some examples of really faces for, I can't really say humans because these are people that really did not exist. They're not there on, they have never been there or been there on Mother Earth. These are images that's been designed by AI. They look very much like real human because AI has been trained on millions of images of faces to come up with these novel ideas. And what we basically, that happened about, I think, 7 years ago when deep learning came to place and when computational resources were strong enough and were cost effective enough for us to run deep learning models on a very large scale. And that basically got these machine learning models to work very efficiently. So people start thinking about ways to scale those methods to apply also in chemistry. So how can we also train those models to learn from the chemistry that we have created and then be able to create also drug-like compound that we human haven't designed or thought of before. So in the background, you probably know that machine learning is in a very abstract way is a kind of it's a very efficient way to capture very complex mathematical functions, really. And because now we have the capacity to build those models on a very large scale, so we have models that has billions of different parameters, we can capture really, really complex mathematical functions. And they've been used both for prediction, so you can predict, for instance, what's in the image by training those models on labeled data. And they can, as I mentioned, they can be used for designing novel ideas or for creating novel things from what we've seen in a more of like unsupervised way. So in chemistry specifically, the way they've been used is basically to train those models on the chemical library that we've created, and then those models learn by trying to reproduce those chemical libraries. And by doing that whole process a number of times, what you get is what we call a latent space or latent space that basically represent the space of chemical that both exist, but also that we can sample from to tap into novel ideas. Now, as you know, machine learning or like deep learning in general started to work when the amount of labeled data went beyond kind of certain thresholds. So when we had labeled data in the ranges of millions, we moved from conventional machine learning or like statistical models to kind of with more data, we moved into shallow neural networks, neural network that has a number of deep layers, into kind of more deep neural network that has billions of parameters. And at the point where we had millions of labeled data, we basically get model that can almost be as efficient or like more efficient than human in labeling images for certain tasks. In chemistry, it is the situation is a bit more tricky because you don't really get that much data. If we just kind of translating existing machine learning models into the chemistry world, then what that basically means is that you need knowledge, obviously, about the drug and like the target, the protein that or the disease that you're targeting. You need at least hundreds to thousands of labeled data points on that specific disease to be able to feed that information into machine learning design and start designing novel ideas. And obviously, you need a lot of activity data to kind of learn more about how certain drugs interact with the different proteins in the body and with our complex biology in general. Well, obviously, we don't know as much both about chemistry and about biology, and that's what makes, I think, discovering novel chemistry and really studying biology very exciting from a research perspective and also from a product perspective, because we don't really have, I would say, the huge understanding. We don't really know how the human body works, and we don't really know how different chemicals interact with our body. So what we've done when we started really the company is basically done the simple analysis, just plotting the number of compounds that we, as a human, designed over the past decades, really over the kind of the past centuries of humanities, and analyzing those against the number of targets or like the targets or diseases that we know of. And we plotted that in this figure that you see here. So what you have here is basically on the x-axis, you have the different targets or the different proteins in our body that we know are disease associated. So we know that these targets are causing, are in the pathways of causing certain diseases. And on the y-axis, you see the number of compounds, but basically projected in a way that these basically the number of compounds on each target. So which compound has been reported in any study that's been reported in the literature on a specific target. And as you can see, the distribution is very skewed in the sense that you have very, very small percentage of the targets where there's a lot of work. There's been a lot of studies, a lot of chemical structures on them. And we have a lot of targets, pretty much more than 80% of those targets, disease associated, but still there is not a lot of study, not a lot of understanding about how we design chemistry against those. And what that basically meant for the past 4 or 5 years is that a lot of the AI in drug discovery efforts have focused on the first category. So targets where there's a lot of information, where we can pretty much take that information, feed it into machine learning models, build model, and then go on and generalize and come up with novel ideas, which is fine. And that within kind of just the past year has led to really kind of fruitful ways of discovering novel chemistry on very well understood target and very well studied diseases. Basically coming up with novel ideas about designing better drugs for those diseases. But going forward, I think the gap that we need to fill to realize the full potential of AI is finding ways to scale AI to be able to work with less and less data, because that is the nature of biology. That is the nature of chemistry. We can't really go on and test all the drugs on human or on animals. That wouldn't be ethical and wouldn't be possible in the first place. So the amount of labeled data usually in this domain specifically is not as much as an AI or machine learning expert would like it to be. So we have to just deal with the fact that we won't get enough data. The data is going to be noisy and sparse and just simply not enough. And we need to find a way to scale AI to deal with those use cases. And that was really the promise where we started Glamorous AI. And that's basically from kind of the fact that the world is missing out on more than 80% of targets causing diseases simply because we don't understand them well enough to design chemistry for them. So in Glamorous, our vision is really to change the face of medicine discovery by making computational methods, AI in general, less reliant on data. And by doing that, we believe that will increase the space of targets that we can address to the 100% of targets causing diseases. And the way we do this is basically, again, by scaling AI to be more data efficient. So this is an example of a very challenging target called KEAP1. It's challenging because, as you can see at the top right picture, this is the 3D structure of the protein. It has a very shallow D pocket, which makes it very challenging for small molecule drug design. It's been there for like a number of years now. We know a number of pharmaceutical companies working on it. There's no drug that targets this protein in the market, but yet it's indicated in cancer, neurodegeneration, and inflammation. So it's really kind of a target that we know is causing a number of diseases, and we haven't managed to crack as humanity yet. So we decided, based on our approach to kind of be more data efficient, to start from that crystal structure, that 3D model of the protein, and just one data point, and build machine learning model that allow us from that one data point to kind of build a chemical space that is relevant for this target, and that allows us to discover novel chemical entities that would potentially lead to drug candidates, to cures. And the process is pretty much kind of, just want to see if this is going to work. So you build a generative model that basically informed by the protein structure, the 3D model of the protein, the kind of the binding, what we call the binding area, how the shape of that binding area looks like. And then by feeding that information about the binding pocket into the model with our starting point, our AI can start generating novel ideas about chemistry, basically, and about designing novel chemistry for that specific target. And by simulating how those drugs interact within the pocket, we can filter, analyze those compounds, prioritize the ones that are likely to work by the end of the day in the lab. I'm just gonna move on to the next slide. So we're now applying this method on a number of different ways. So the one way is the kind of example that I mentioned, and within that kind of example, we ended up generating a number of ideas, filtering them, going through the process of like filtering, analyzing those compounds, understanding the interaction with the protein, and we prioritized only 3 compounds. We send those to the lab, and one of them is active. This is a huge reduction in terms of the timeline to get to a small molecule inhibitor. So we did that whole process in under 6 weeks. And we tested only 3 compounds. And usually, kind of in a traditional pharmaceutical company, you would need to test hundred thousands of compounds to get to that point. So we tested a kind of a number of different other approaches on different sizes of data to kind of just validate our ideas around building a scalable machine learning model that are more data efficient. And what you can see here are a number of examples, for instance, taking from tox data, so basic toxicity data reported on from tox data on humans. So when you test compounds on human, you get like tox values. And you get that kind of labeled data, and you train machine learning models on. And I think the general assumption that machine learning, to train machine learning, you would need larger and larger and larger data sets. And what we believe in is that you don't really need larger data sets. You need kind of more informative data sets. And what you can see here is the proof of that. So we trained different types of machine learning models. The one that you see in red are the ones that are trained by our approach, which are basically optimized for data efficiency. And then the other one that you see in blue are the kind of standard machine learning approaches. And what you can see here is that with our approach, our model achieved the required accuracy, basically using significantly less data than standard approaches. So like the ideal scenario would be like using just 20% of the data to train the model to reach the optimal accuracy. And in other scenarios, we get like about 50% of the data. And what that basically tells you is that you just need to be smarter when it comes to collecting data, rather than just kind of randomly labeling and randomly curating those data. And by doing that, you basically do less experiments, you reduce the time that's required, and you still achieve the same level of accuracy as you would by collecting huge data sets. As I mentioned, the core kind of mission in building Glamorous AI was to be able not just to build that amazing technology and scale it, but also to be able to provide it to a wide range of drug hunters and experts to get access to the technology and utilize it in their own drug discovery programs. And that's why we're very much focused on building a scalable AI platform to support that kind of process. So at the moment, the platform has what we believe is the largest repository of state-of-the-art AI models for prediction of many different types of activity data from chemical structures. Our models are trained on one of the largest repository of publicly available data sources. So it's about 10x bigger than what people usually study when it comes to applications and research purposes. We build it in a way that allows drug hunters and non-machine learning or non-data scientists to really intuitively interact with the interface without having to have a lot of coding experience or a machine learning background. So it's just a matter of uploading data, visualizing, manipulating the data, and then running machine learning processes without having to worry about the cleaning and the kind of spinning out AWS instances, or like building those models and optimizing those and basically validating the accuracy of those models. So we do all of that in the background, and all you care about as a user or as a medicinal chemist is feeding in the raw data, visualizing the results, and feeding back your input within the interface to get better and better models. So our models is, generally speaking, can be trained to the same level of accuracy as the state of the art or better, using at least 50% of less data. And we have a lot of the models that are at the moment surpassing the level of accuracy of the state of the art. And we care not just about accuracy, but also about generalizability. So our models can generalize and can go beyond training data and predict as well on unseen data. So I want to stop there. Obviously, we're going to have a discussion around kind of what we're building and why we care about these problems, and we're happy to answer any questions around those. But going back, I think, to the panel discussion, I'm very much interested also in hearing from the other panelists.
Yeah, Noor, thanks a lot for this great overview presentation, both like what the initial problem is and what the starting position is and where your company is headed. I also like the autoencoder and the generative approach, a reference to NVIDIA.
Yeah, thank you.
I wrote down some questions. We will dive into these later, because now for the second keynote, so see you in 12 minutes. I now take you off stage. Yes. And yeah, now actually it is a very special pleasure for us, because during the preparation of the 5th State of AI event, we found out we never had the Austrian startup on stage. So why was that the case? I don't know. But it's just by coincidence actually that during the preparation we found out there is actually one very well-funded startup, Celeris Therapeutics, in Austria, dealing with the topic of tonight. So with me on the stage, I welcome Noah Weber, the CTO of Celeris. And yeah, please, Noah, if you could spare some words or make a brief intro, and also please do not forget to share your presentation, because in the backstage I see it vanished.
Yes. Can you hear me?
Loud and clear.
This is perfect. Can you see my presentation?
Yes, now we can.
Okay, perfect.
So here it is, perfect. Great.
Yes.
So yeah, I will tune in when it's the end of your presentation again.
Right. Thank you, Clemens. Yeah. So basically, as Clemens said, we are Celeris Therapeutics. We are a startup company from Graz, Austria, and we are trying to tackle something similar that Noor was talking about. She was specifically alluding to the 80% of the pathogenic proteins not being able to target it, and we believe that we solved that solution using degrader technologies. But basically, the year is 2021, and we are still unable to treat Alzheimer's, Parkinson's, Huntington's, and many other diseases. The question becomes, why? Well, 80% of all of the possible pathogenic proteins, meaning the proteins that are associated with diseases, are undruggable. The less than 20% are inhibitable, meaning they can be targeted with other molecules. But to tackle the rest 80%, the larger portion of this set of possible molecules, one needs to have another approach other than inhibition. The possible approaches there, or the therapeutic interventions, are either gene-based, like CRISPR that we heard all over the news, or RNA approaches, or direct inhibition of the pathogenic protein in case. But alternative to directly inhibiting the protein is degrading it. Now, this is maybe a little bit complex, abstract statement from biochemistry. And what does actually degrading a protein mean? Well, degrader moves the topic or the focus from inhibition to degradation. That means that we no longer have to fight the symptoms, but we eliminate the cause of disease from the get-go. Now, this picture may sound a little bit complex, or there's a lot of moving parts, but what I want to achieve with this is to just motivate how do we move from this biochemical technology, degrader technologies, to the AI or machine learning space. So if you look at this picture, you will see on the far left, there's something called the E3 protein and the small bridge in between, and then the POI, the protein of interest. Now, when we find this bridge in between that allows us to connect the E3 and the POI, basically we can then, with the help of the E3 protein, start off the degradation of the protein of interest, the protein that's associated with the disease, the pathogenic protein. And if we do this correctly and successfully, we eliminated the cause of disease from the start. Now, looking at the far left picture, there we see that in more detail, there is not only these two molecules, the E3 and the protein of interest, but there are also some smaller molecules, meaning the E3 ligase ligand, the linker in between, and the protein of interest ligand. So all in all, you can say that we are talking about 5 molecules, some bigger, some smaller, that need to interact together to basically generate this degrader technology. Now the question becomes, how do we translate this to the AI domain? First up is the data. The data space of the possible molecules is huge. Some sources say it's as the number of atoms in the universe, but we are talking about a huge space of data that cannot be easily parsed. Now that we have our data and we know what we want to talk about, we also need to talk about suitable representation. So what is a native representation for our molecules? One thing that's very peculiar about this is that it doesn't suffice to represent data or the molecules at hand as there is, but we also have to model their relationships, intermolecular and interatomic relationships. The type of representation that's very suitable for this that we found out is the graph representation, where every atom in this particular molecule can be represented as a node, right? And every relationship or every feature can be represented as edge. And then we have this non-Euclidean representation that allows for a lot more complex interdependencies between the data. And then it allows us basically to learn more things that then translate to making better predictions regarding the previous ternary complex or the 5 molecules that I mentioned. Another enabler, and that translates this actually to the AI domain, is figuring out smart algorithms that can work on these graph data. So there we had to pool basically different disciplines that allowed us to tackle these problems on huge dimensional, so a big set of graph data. And these are the different disciplines of geometric deep learning, generative models, active learning, right? So we had to parse the huge space of data smarter, and Bayesian optimization, where we needed to find the subsequent steps in our decision for a molecule smarter than just a brute force approach. I wanted to linger for a moment on the importance of the non-Euclidean data. This picture, and for all of our computer vision experts that could confirm, if you were to train a standard convolutional network, most likely would we classify the first cat and the last picture of a dog together, just based on the simple, naive 2D representation of pixels. But the relationship is a little bit more complex than that. Obviously, we see that the actual thing that needs to be grouped together are the first 2 pictures of 2 cats. The reason being is that their relationships are a bit more complex than just the pixels at hand, right? They have their hereditary, they have their species features, they have more complex interconnections between them than just what's to be seen on the screen, on the pixels that represent the images. Now, what are tricky with the non-Euclidean data is that Euclideans, not everything can be modeled with the Euclidean similarities, as we saw with these images, right? So if we have images that are just similar in the color but not really similar in other properties, then we won't be able to make these classifications. Also, the relationships, right? As I noted, in the sense of the cat and their hereditary or species properties, but also if we talk about molecules, right? The molecules are not interacting in vacuum. All of the intermolecular and interatomic properties and relationships also need to be modeled into data, right? And finally, if we even manage, and this is already possible since for a couple of years, the research in the geometric deep learning has been extensive. If we manage to model this, then the standard paradigms of deep learning or machine learning are no longer applicable. I'm going to give you one super simple example. Convolutions as a convolutional operation on a standard data, it needs to have a motion of left and right. So where do I shift or move my convolutional operator across the images? Now, this motion of left or right or orientation cannot be directly modeled in graphs. So the convolutional operator needs to be generalized in order to operate on the graph data. What were other AI enablers that allowed us to solve this problem of targeted protein degradation? First of all, data quality and quantity. We saw just the recent news was the AlphaFold, where they didn't only develop the huge deep learning pipeline for predicting the 3D shape of the proteins, but they also open sourced all of their predictions, right? But in the previous years leading up to that, the quality and the quantity of data describing these molecules has really grown exponentially. Other points that helped us a lot is the active learning research. So the research in the field of active learning, enabling querying and parsing of huge sets of data. So we cannot query something that is big as the number of atoms in your universe. We need to have smart query approaches, smart algorithms that allow us to pick our data points smartly and wisely. Furthermore, the new methods in chemobioinformatics. So we are not only leveraging purely machine learning, but also research that's being done there. For example, docking approaches to specify how good a certain pose or certain connection between two molecules is. Then, obviously, as I mentioned, the graph deep learning. This is something that has been highly researched, and it's been a big hot topic in the last couple of years, where this non-Euclidean data is actually all around us, and we need smart approaches to also act upon that. And then, obviously, the standard one, which just continues to be a huge enabler, is computing power and cloud, right? So the NVIDIA graphic cards and all of the other powerful TPUs, GPUs, and whatever allow us to crunch the data super fast, which is needed. Again, the algorithms that operate and the data that's here, it's just huge. The standard CPUs are just not going to cut it. And final thing that I really believe was essential for us to solve this problem is interdisciplinary work, right? So if we were to approach this problem very naively and say, okay, we are just a group of machine learning researchers or data scientists, and we are going to just get the data and then try to mix it, try to do something smart, and then hopefully get the desired predictions, this would most likely not work. What we were very careful about in Cellaris is making sure that our experts in chemistry, biology, mathematicians, engineers, and researchers work together to translate the requirements of their own disciplines into data, so that it doesn't happen that we model something with data with the requirements that weren't there, and only at the end we decide some physical property was not preserved or some chemical relationship was not there. So it was really crucial for us to actually tackle the targeted protein degradation with the help of our scientists from all of these different disciplines. And yeah, that was Cellorist Therapeutics, where we are going to make better drugs faster.
Great. Thank you very much, Noah. I also showed just now a banner for those of you that want to know more about Cellorist. You can always head over to their homepage or use the QR code in the stream. Yeah, with this, thanks a lot. And I would like to directly move over to the podium discussion. So with me on the stage, I welcome now Noor, again, keynote speaker 1. Then John Cumbers. Hi, John. Can you hear us?
I can,
yeah.
The
microphone is working in the first try. I'm not used to this anymore. And last but not least, Stefan Sauermann. Okay, now all the microphones are working. Yeah, before diving into the let's say panel discussion, maybe one general comment from my side. When putting together such a panel, we always want to make sure that we have lots of different viewpoints, but also people representing different stakeholder groups. I mean, for Noor, it's obviously the R&D driven, deep tech driven startup founder. In the person of Mr. Sauermann, it's the regulatory view, because he is also involved in the IEEE, and this is as much regulatory and standardization as it gets. And last but not least, John, who has a background in synthetic biology, even worked at NASA before, so not aliens, but synthetic biology on Earth, and who is the founder of SynBioBeta, which you can maybe tell us a couple of words about.
Sure, happy to. SynBioBeta is a global network for synthetic biology entrepreneurs and investors. We are known for our annual conference and trade show, which is coming up here in the Bay Area in 6 weeks' time. And it's actually our 10-year anniversary. And we have been tracking the growth of the field of synthetic biology for the last 10 years, seeing it grow from $250 million a year when we started in 2012 to now we are predicting $15 billion will be invested in the industry in 2021. And before that, I was at NASA, as you said, for 7 years, working on food production and waste materials and processing. And I have a PhD in cell molecular biology from Brown University and a master's in bioinformatics from Edinburgh University.
Excellent. So since it was mentioned in the previous keynote, I directly want to use this also as an entry to the questions I prepared, because I remember 4 years ago when we held the first event, the memory of, on the one hand side, autonomous driving was still very fresh, or so-called autonomous driving, and especially AlphaGo and reinforcement learning, the technology behind it. It's actually a coincidence that back then it was Google that was making the headlines. Now with AlphaFold, it's again Google making the headlines. But with 2020 hindsight, I would say reinforcement learning has lots of promise, but it did not lead it to sudden applications in the industry. It's rather still gradually coming, like in trading systems, in logistics, and so on. But it's not everywhere. And this would be a question to you, Noor, and John. How representative are results like AlphaFold, for example, or is this as far away as reinforcement learning and AlphaGo was 5 years ago?
Sure, I'll go first. I think that AlphaFold cannot be underestimated, the impact that it has already had on the field of biotechnology. I have a good friend of mine who runs an editing business, and he reviews and edits a lot of papers for Nature and Cell. And he said that overnight, nearly every single paper that he is seeing is now referencing AlphaFold and using structures in AlphaFold. We're not even seeing this yet in the literature because they're going through the review process. But he said it's just like a sea change. It's like the invention of BLAST, or he even described it as the discovery of the structure of DNA as to the significance of it. So I think it's difficult to look at the broader set of applications of AI to biology, because there are many different various technologies out there and companies out there yet to be proven that it's working. But just in the single case alone of AlphaFold, I think it is a sea change in an amazing application of AI to do an amazing amount of good for not just drug discovery, but all of the field of bioengineering.
That's quite a statement to put it on the level of the DNA. I hope they write a book about it, like James D. Watson with double helix. That would be cool.
Yeah, that would be awesome.
Yeah, I'd probably like play the devil's advocate here and say, I think it's still a bit early to start kind of like there's something that's called like the Lindy effect, or like Lindy compatible kind of events, which basically the event that pass the test of time. And I think for AlphaFold specifically, it's still early to kind of see whether it's gonna be like that big moment where we're gonna solve like crystal structure of protein, like for all of protein, like in a human body, and then kind of just claim that this problem is solved, or we're gonna like we solved a big part of the problem, and then over the next year we're gonna see like what are really the gaps and what are really the boundaries of it before where it's gonna succeed and where it's gonna struggle. I think it's gonna be a very interesting kind of things to see unfold over the next few years. It's definitely kind of a big moment in time in terms of seeing computational methods actually and AI actually fruitful and solving real problem that's been there for decades. But I think we shouldn't be kind of in the mindset of like making big claims and just saying that the problem is solved and now we should move on. I think that's kind of a lot of academic institutes now are making kind of a shift in mindset in that they are now adopting AI. They're more excited about AI after seeing AlphaFold. But I think we also see a lot of efforts in terms of like proving and validating where it's going to work and where it's not going to work as efficient as some expect.
Thank you. This brings me actually to the second question. I mean, you showed the generated faces, and I think GANs are like one of the prime examples how much compute power you need to really create something of high value. And if you look back in the last decade between AlexNet and I think it was not AlphaGo, but AlexZero, not AlexZero, AlphaZero, AlexNet to AlphaZero, you had the compute increase by a factor of 300,000. So I remember 2 years ago, the guy from Springer was saying that especially in the field of NLP, you're basically hitting the wall because the models become larger and larger, and these very large models, I think since yesterday are called foundation models by Stanford. They are good for these very large institutions, but for startups, it's basically impossible to really act in this field anymore. How is this situation in biotech or drug discovery? Do you think that you will soon need to switch to custom silicon, let's say GraphCore, FPGAs or whatever? Or is there still lots of road ahead where you don't need custom architectures?
I think with AlphaZero specifically, one of the main hurdles that blocked wider adoption is the size of the model that basically left everyone like wondering whether like is it just for Google because they are the only one with like super servers and super like hardware that can run it. Software like hardware is moving in the pace that is, as you mentioned, significantly enabling in terms of AI for drug discovery specifically. We haven't seen really like for us specifically the need to like extra large servers or HPC powers to kind of run our methods. I think that the size of the models, given specifically that the domain that we work with, there's not really a lot of data in the first place. So our job is mostly around managing small databases and getting AI to work on those. So we're not kind of, at least from a small molecule perspective, we're not as HPC heavy as probably others who work in more kind of biology and protein structure solving problems.
Great. Now after the initial part was, let's say, about the state of the technology and what to expect, also is AlphaFold a watershed moment or not, I would like to come into an, I wouldn't say more interesting topic, but if you talk about biotech and AI, obviously regulation is the big elephant in the room, especially the EU just announced published the AI Act, which should come into force in about 2 years. So there's lots of discussion about it. And for this very special reason, we also welcome Stefan Sauermann with us on the stage. So maybe Stefan, you can make a very short intro. What is your work and how does it relate to AI and biotech?
Yeah, thank you. I'm very happy to be here because like my work is here at the university and in our research is in e-health and in things like electronic healthcare records, telemedicine, and let's say transforming the healthcare enterprise into digital. And so you're applying knowledge on individual patients. And the knowledge in medicine is increasing, and we've seen 2 presentations which are dramatically increasing the knowledge, and there are many other researchers which are doing the same. And this definitely challenges the medical professions significantly in really making use of that knowledge. And so we need AI to really harvest the benefits from all of this work. And different forms of AI have been around for decades. Like there are rather simple expert systems. There are expert systems around which are also in place as medical devices and which are regulated. And we can go very deep here if you wish. My role is, I must say honestly, I'm not really a dedicated, experienced medical device regulation expert. I'm teaching this space and I have the overview. There are others who understand much deeper, who would really help you to make it through regulation. So I'm looking at this from a rather high-flying view. Yeah, that's me.
Great. Then I would directly also direct the first question in the regulatory area to you. What is in your opinion and in layman terms, what are the biggest regulatory differences at the moment between the blocks? So especially between Europe and the United States, because this is where most startups in the ICT sphere are created.
Yeah, as far as I understand, we have regulation in place with the target to assure a useful level of safety. So you're bringing in technology, you don't want to harm anybody. And the examples we've heard today, I would consider are not treating patients directly. So I would not believe that they are regulated. At least that is my view. In other applications, like you have, for example, AI which analyzes X-ray pictures of the individual patient to detect cancer or detect damage of bones, then that would be a medical device. And you want to reduce the risk of harm to individuals and also to organizations. Now, you can't negotiate safety. Products have to be safe. And that is the goal of regulation. And my feeling is all over the planet, you have well-defined safety standards. So we know what safety means and how to establish it. And I would believe that this is similar between all the regions. And then on top of that, you are then introducing procedures, application to put a device to the market. And I think that that is where the differences are coming up. How much support is there for an individual company to bring a medical device to the market together with the authorities and experts to assess the device and to answer a lot of questions about risk management. And I think that some regions are more supportive and others are more complex in the procedures. And I would believe that this space is actually where the differences lie.
Okay, and this would actually one question to the entire panel. On the one hand side, with AI, there's lots of competition when it comes to R&D labs, or let's say startups, that they create the perfect boundary conditions. So think of testing grounds, or let's say testing tracks. This was especially in the United States, a game that many states played against each other, with Arizona and so on. But also, when I think of biotech, I think the previous administration in Japan made big efforts to get become more attractive for gene editing, for example, than other fields. Is there something like a competition going on between the different regions that you see, both from investment perspective or startup perspective?
Yeah, go ahead, Noah.
I think generally speaking, it seems like AI, like speaking of my kind of the ecosystem that I'm within, like AI for drug discovery seems to be attracting a lot of attention globally. Like we see a lot of startups in the US, we see a lot of startups in Europe. Similarly in China is moving very fast, both in terms of number of publications and research that's happening in the field, but also in terms of the startup and the service offering. Having the advantage of course, of like also having a lot of funding going into that kind of Chinese market, and I would say easier access and more kind of open market, both in terms of like the pharmaceutical industry that is out there and access to the kind of the startup ecosystem and so on and so forth. So a lot of advances are happening there, and I see a lot of startups also kind of opening subsidiaries or like headquarters in China specifically as well. Probably the Asian market and the Japanese market is still a bit behind in terms of the number of startups that are coming in, but they're also very strong in terms of just the market and the being open to adopting new approaches and new technologies.
And how is this from an investment perspective? Do you also see that different types of startups are funded? So, I mean, if I make a very rough analogy, in Europe, especially in the DACH region, you see lots of startups in the field of production, manufacturing, and logistics. There's basically almost nothing in the field of B2C. So, if you think of generative networks, something like Rosebud.ai would never have been created in Europe. That's the typical California startup. So,
yeah.
Are there also differences in the drug discovery and biotech sector that you see?
I think there's a lot happening in terms of the therapeutic modalities, like small molecules versus like regenerative medicine versus like biologics and so on and so forth. There has been a lot of investment in the sector over the past few years, and I think now it is in the phase where it's kind of like probably at a tipping point. There's a lot of startup in the ecosystem at the moment that I see over the next few years are kind of probably be less investment and more kind of giving those companies the time that's needed to kind of either prove themselves and kind of pass into being sustainable companies and going kind of to be bigger. It's hard to tell. There's like now like the number of startups in the space is in the hundreds at the moment. So there's no doubt there is a lot of innovation happening and lots of startups coming, and there's been a lot of investment in the field. I think now it's more like improving the technology, bringing it to matureness, seeing it through kind of to preclinical and clinical studies, getting the first AI drug discovered compounds to patients. That's definitely going to take some time. So we'll see. The next few years are certainly going to be so exciting.
Yeah, but what's your take on this one, John, like the US angle?
I think coming from the UK, I have a European perspective, but living in California now for over a decade, the US really has the greatest capital markets in the world because they have this culture of feeding it forward, particularly Silicon Valley, reinvesting in the next big thing. And what we're seeing here in Silicon Valley over the last decade is a reinvestment from tech founders into biotech companies. And bringing that comes more than just the capital that they bring. It also comes the expertise in machine learning and AI. So we're seeing this really healthy IPO market. Companies like Recursion, companies like Schrödinger, AbCellera, Berkeley Lights, they're all at the intersection of technology and biology. And that's what I think is the most exciting area. And we're seeing a healthy capital market from everything from pre-seed up to IPO. And as I said earlier, we put out a report at SynBio Beta predicting 15 billion invested in the field. That is broadly in synthetic biology. We haven't done anything specifically on AI. But I'm also seeing great signs of investment from the UK. Great folks like I have friends at Leaps by Bayer in Berlin and Blue Yard at Berlin, and they're all looking at digital transformations and AI. Amadeus Capital, of course, with Hermann Hauser in the UK, and Sofinova in France, and a few others. So I'm very excited by what I'm seeing on both sides of the Atlantic, and I think COVID has ushered in an acceleration of biological technologies, and many of us have been talking about the century of biology, and I really think it has begun. So I couldn't be more excited about all of the tools and things like the company that Noah's working on, and all of the other tools and technologies that are becoming available, and this confluence of technology and biology, where the new startups, the new founders don't need a PhD anymore. They can grab these tools and go run with it and start a company. And we're seeing that with accelerators like Y Combinator and IndieBio and many others that are just fueling this new generation of combination biotech and informatics and AI startups.
This also solves the famous double PhD problem that AI startups always face. That you need to have a PhD in AI, or lots of PhDs, plus the PhDs of the respective domain. When it comes,
just to give you an example of the capital, I heard just yesterday that Google had recruited somebody with an AI PhD. They just finished their PhD and was paying them $800,000 a year.
This should cover actually the cost for the kindergarten and the real estate in the Bay Area. Maybe one soy latte per day.
Yeah, that's true.
No, but these are amazing amounts. I'm just reminded by the organizers that we should come to an end, unfortunately. So maybe one final question where I would start with Stefan, but I would also like to get the feedback from Noor and John. When it comes to the regulatory view, the EU is thinking lots in terms of sandboxes that you use, for example, for fintech or autonomous driving are the obvious candidates. But these are, of course, just one possible way to approach this. What would you wish for or expect from regulatory bodies to help with the acceleration of the development of biotech startups?
I think a good example is the COVID vaccination. Like Israel has been very early and has been actively engaging with manufacturers to provide data out of the early stages of regulatory processes and to use the vaccine very early in the process, much earlier than Europe. And it's a giving and taking between the community, which vaccinates, and feeding data back to the manufacturer into the regulatory processes. And this has not been done, let's say, in the European Union. So they waited until the data came from somewhere else. And I see a very positive movement in Europe, the European data spaces, and also in Austria, legal discussions are in place to provide process data, let's say from medicine, from e-government, from many domains, under very controlled conditions of pseudonymization, anonymization, privacy, to research partners and also to industry. And I believe the vaccine in Israel is one example for speeding up innovation. And I think what the European Union is now doing with data spaces is going into the same direction. And I believe what you mentioned, sandboxes and other types of support, better support in the formal side of regulatory processes, peer support. So there are many ways you can support companies with, and I think sharing data is one main element of that.
Good.
Just,
John?
sorry, John. Go on, John. This is yours.
Yeah, I would. I think you've got to be very careful with regulation, because I've seen it clearly with synthetic biology that you regulate an area and founders are not excited to work in a regulated area. This is actually, if you look between biofuels and biopharma, which are both regulated or subsidized. You know, the biofuels market is quite skewed and subsidized. The biopharma are extremely regulated. So both have quite a lot of government intervention. You don't see a huge amount of innovation there. But if you look at all the stuff in the middle, consumer applications, food products, which have some level of regulation, but not a huge amount, materials, chemicals, software products in biology, all of that stuff flourishes, and there's a healthy early stage startup ecosystem. At the other extremes, they flourish, but they flourish in different ways. The biopharma incredibly skewed towards high valuations and expensive development costs, and the fuels market, the biofuels market, heavily skewed, heavily at the whim of government regulation. So I think that regulation is good for our economy. I think it's good for the health and safety of people and the environment. But I think it needs to be used with as a very careful tool, because as soon as you regulate, you turn off a whole lot of entrepreneurs from innovating in that space.
No, I think I just want to echo what Stefan and John mentioned. I think regulation scares both founders and also investors. I think that's why it scares founders, because as soon as you go into regulated area, the likelihood of like investors investing in that area is going to be definitely smaller and like thinner. So we tend to kind of steer away from those, which is not a fault of ours. I think it's just kind of how things are. But I think just echoing what Stefan mentioned, COVID certainly has kind of accelerated and opened some eyes around what needs to be regulated and how we should be thinking about it, because it also increased the awareness around the value of data and the value of giving that data to innovators early on, because without access to data, you can't really innovate. And it's been a big hurdle here in the UK, for instance, with the NHS and access to data from the NHS. That's blocked a lot of innovation. And now people within the NHS are thinking about ways to kind of open innovation and how we share without having any privacy, anonymous, solving all those issues and then sharing the data. So it's a big, I think, thing on everyone's mind. We do know that data is gold mined, and we know that we do have the tools to mine that gold. It's just a matter of combining the two and removing the hurdles from the way.
Great. So I'm reminded again that we reached the end already 10 minutes ago, but still, thanks a lot for the interesting insights. Also maybe not unimportant for the three of you, if you. All of this is live recorded, so if you want to share the stream afterwards, just CNP or directly share the LinkedIn feed. With this, I would like to thank you very much for joining. It's too bad that thanks to COVID, we could not meet in person, because normally it always takes place in the Alps in Tyrol. But maybe, who knows, maybe you will find a solution until next year and we make an offline event again. So pleasure to have you, and looking forward to see, maybe meet you one day.
Thank you much. Thanks for the invite, and great meeting you all.
Thank you. Bye.
Thanks, Clemens, and thanks, Clara. Look forward to seeing you in Austria sometime soon.
Same wise. Thanks. Bye-bye.
Bye.
Bye.
Great. Yeah. And this brings us to the end of this year's edition. As I mentioned, the video is still available for watching afterwards. And last but not least, and I show this banner for the very last time, if, as Clara mentioned in the beginning, if you would like to see the pitches of this year's summer patch of the ISCT, just head over to network.isct.at, log in, create an account, and yeah, enjoy the show. So with this, I wish you a very nice evening and see you soon again. Bye bye.